So good afternoon, everybody, and welcome to this very exciting discussion. I'm Judith Molnár, VP of Translational Development at Neo4, which is a small immuno-oncology biotech company, which is my day job. I'm also a One Nucleus board member. And my work really focuses on bridging scientific innovation with regulatory and clinical development. And today's topic is a very exciting one for me to discuss. And I'm delighted to chair this panel. And before we dive into the discussion, I'd like to ask everybody to tell me a little bit about your background and introduce yourself. I know people know your name, but if you want to add, I give you the chance.
Yeah, thank you, Judith. My background is in tissue engineering. I have trained all my life creating the most complex human model ever to the degree that you can implant it. So I gave up on the implanting part, but everything I have learned on the tissue engineering field during my PhD at Molly Stevens, I have applied it into the practical drug development application. And I think hopefully we will create tissue engineering applications one day, but all of our learnings from here is already making an impact, as you see, and I really like being in the middle of it.
Thank you. Leona.
Hi, everybody. I'm Leona Fitzgerald, and I'm the Global Head of Regulatory Affairs for a CRO, PPD, part of Thermo Fisher Scientific. In our business, we are involved in supporting sponsors, drug developers, in moving their molecules, both devices and drugs, through the clinical pathway into licensing and then post-licensing. One part of my role as well is I'm lucky enough to be engaging with regulators to help set the scene. What does industry need? What does the stakeholder network look like and need, and how does that feed into practical development of legislation and policy? So we're often at a seat at the table talking about things that there is no guidance on, and what does that early guidance look like? An example would be how we had to spring forward very quickly during the pandemic and decentralize everything, and how we could maintain patient safety, quality, but also data integrity through the development pathway.
Yeah, I'll go. It feels weird to introduce myself after I've already spoken to you. Hannah, my background really is as an epidemiologist, so that's the angle I come at clinical trials from. I've worked on massive kind of phase 3-4 clinical trials when I was at the University of Oxford, CTSU, and then more recently actually at Benevolent AI, we ran a couple of clinical trials there as well, so it's kind of both ends of the spectrum there. I spent a lot of time early on in my career working on electronic patient record systems, so actually in hospitals with all that clinical data. I've seen how far we've come with that, which is amazing, but I've also seen the impact of everything else that goes on in people's lives and what that has on their health outcomes, and also, as a result, trial outcomes.
So hi again, everybody. So I'm Tom Koszczewski, the Chief Scientific Officer at CM Bio. So as I alluded to in my talk, I'm an immunologist by training, did a PhD in Imperial College London. I helped set up a spin-out company there called NKIO, which develops NK cell therapies. Also worked at GSK, like many people around here, in Stevenage for a period developing monoclonal antibodies. Joined CM Bio over a decade ago when we were just a small little startup with a tiny little lab, and have helped grow the business and lead the business ever since. So I'm a biologist, but also a bit of an engineer by osmosis by working with our amazing engineering team.
Thank you very much. So R&D is under increasing pressure to be faster, more predictive, and more efficient. So that's not new. However, at the same time, scientific innovation is challenging some of the assumptions that have underpinned the regulatory and the clinical development paradigms for decades. So for example, from AI and NEMS, or new approach methodologies, to adaptive trial designs and novel regulatory pathways, we are seeing opportunities that potentially fundamentally changes things, how we've done, how medicines are being developed. So today, I'd like us to explore what these changes mean in practice, where are the opportunities, and what still needs to happen before these innovations really become mainstream. So I'd like to start by exploring a fundamental question, that are regulatory and development paradigms really changing? It was mentioned many times that last year in April, the FDA announced plans to phase out animal, not just toxicologies, generally animal testing, particularly for monoclonal antibodies. And they are really encouraging the use of alternative approaches such as AI models, organoids, and computational methods. So this feels like a really important signal from regulators, but how far are we really from a world where these approaches really replace traditional methodologies? So for example, if a company wanted to replace or significantly reduce traditional animal or clinical development steps using NAMs or AI-enabled approaches, what evidence package would regulators need to see to be confident in those decisions? So maybe I'm looking at Leona as the regulator at the table.
I mean, it's a big question. Yeah. I'm gonna start with the classic regulator feedback of it depends. A lot of these things will be, as we've discussed a little bit over the course of the day already, dependent on what your molecule is, what the target organism is, the mechanism of action, whether there's something similar to it already in further down the clinical line, maybe already commercially available, and the data confidence in the safety and efficacy of that product. So regulators will be looking at understanding the quality of the manufacture of the product, the safety information that you can garner through both whatever the non-clinical package could be, whether that's animal models or something else, and then some sort of clinical evidence. Depending on the kind of disease you're in, the clinical evidence will be more or less. So rare diseases, for example, will have a much smaller clinical package purely from the nature of the patient population available. And that's where going into post-approval clinical evidence and real-world evidence strengthens your label claim. So often it's really depending what label claim you want to go forward with first and what is the data to support that claim. Sometimes you can get that, sometimes you have to compromise and have a smaller label and then generate more data as you progress.
Maybe I can add on that based on my experience. So when big organizations, let's say really big pharma, have looked at it exactly with that question in mind, where we can specifically or at most can cut animal experiments, but more importantly can improve the science or predictivity. There are two-way schools of thought on that, and there are different pharma companies who utilize both. One is going really horizontal, trying to touch everything, having multiple efficacy models, multiple safety models, trying to do everything at once for all modalities. And a lot of the pharma companies have started testing MPS systems like that, just because the senior leadership would like to see the highest value and return of investment in the widest possible way. But that at the beginning, when you are just initially getting into that, that is kind of difficult situation to be in because it is extremely resource consuming, and there is an exponential curve where you will start seeing the impact, the return of it. So there will be a long period of time where you will invest a lot and see nothing. On the other school of thought, which is I call AstraZeneca method, you pick one thing that is very...
There are other pharma companies available.
For that school of thought. They looked at what is very important to them at that given time and go with a moonshot project, which was creating a bone marrow on chip. At the time when they have started in 2014-15, that was an absolutely moonshot. And they really invested all of their resources into that, as well as with others, but very heavily into that. And the results and the success they have achieved is absolutely game changer. When it comes to bone marrow toxicity, rats are highly predictive as well as far as the animals are, yet with the results and the tool that's available as a bone marrow on chip, rat experiments are rarely needed now. So I would say, this is the long answer. Short answer is go vertical. Pick something that is very important to your organization and really focus on that. So everything, not try to touching everything in your whole organization. Everything that is in your organization should touch that at some point and expand it from there to make the biggest impact.
And do you think regulators leading the change, or they are responding to the innovation explosion that's really happening in the industry?
Responding. They are responding. I think they're very open to collaborate, and there's been a lot of activity in the last probably 18 months, 2 years, where regulators are asking, what are you doing with this AI stuff? What's going on? How are you using it? And then they're testing it internally themselves, more from a process and workflow efficiency element than anything else. I think there's still a lot of suspicion, and the only way we're going to overcome that is with transparency and opening up, how is it used, how is it learning, how is it validated. So at the moment, they are being very reactive, but they are looking for everyone to help them understand what's possible.
Mm-hmm. I totally agree with that as well. Like in terms of the being transparent, you need the evidence. You need to be able to see why that clinical decision's been made. You need to be able to trace that back and build out that supporting package. So I think, in order to be able to validate it, it needs to be very open. You can't have these kind of black box AI systems where you don't know why it's come to that level of reasoning.
Yeah, I think the thing I'll add is that the regulators have been relatively consistent with their messaging, in that they have never been against the use of new technologies in any regulatory filing. It's about the amount of supporting evidence that you might have to bring with it to demonstrate why you've used a particular tool. So as certain particular tools become more commonplace, then obviously the amount of supporting evidence that is required does start to diminish somewhat. But there is no barrier to any tool being used if it's logical and appropriately used and appropriately tested. And the sort of interesting dynamic is that in every part of the process, the amount of, let's call it validation, it's not always the right word, but that's required is going to vary. So certainly for efficacy testing, the level of validation is going to be lower, right? Because the regulators really care about toxicology. They really only worry about, are we going to hurt a patient or not? They're less worried about, are we going to treat the patient? So on the efficacy side, the barrier to getting any of these new approaches in is much lower. So I think the ultimate gold standard of animal testing and toxicology is still going to be there, but it's about augmenting that with these new tools. So it's going to vary.
Yeah, that's really good points. And so it sounds like that the science is much more ahead of the regulators. So where do you see the biggest gap between science and regulators, or what needs to happen to bring them together?
I would say it is a stakeholder feedback. So I have been part of these conversations at the EMA side, trying to understand the stakeholder feedback, what is feasible on the organ-on-chip side, what we can actually realistically change, and what the end users would like to be using them most often to be able to bridge that gap so that what they update is actually usable. So I guess that is the biggest gap that needs to be filled in, which is in the process of being filled in.
I think also there's a big educational piece within regulators, right? If you take the EMA or the FDA, these are enormous organisations with a lot of long-standing employees that have done things a certain way for quite a long time, and so there's a lot of education to be done for those. And I know that certain parts of those organisations are going through that educational process, but it takes time, and different parts of it will be embracing that education in different ways. And I think it's incumbent on everybody to kind of try and be part of that as much as we can.
So let's take the pressure of the regulators and change direction. I'd like to talk about data, because all these novel technologies, they generate a lot of data. And is it true that nowadays the problem is not to obtain this data, it's more like what to do with all of that?
I think I'd argue that we still need more data. That there's still not enough data. I think human biology is infinitely complex, and that we still don't really understand it. I think that as a tool developer, there's still a huge amount of data that we can't access. There's some good data out there in certain public databases, but lots of the amount of failed drug data that exists within pharma companies' databases is phenomenal. I don't have a magic idea about how we ever get any of it out, but hopefully someone does somewhere. Because I think the more of those examples we can get out there, the more comparison people can make for some of these tools to say what does it have to predict and what does it not. Because one, for example, practical challenge that we have, and all the beautiful array of modalities that were talked about this morning, is you can't go and buy those from Sigma or Merck just off the shelf, right? And then run a screening thing to validate against them, right? They exist in tiny quantities. They're incredibly intricate to make. Some of them, the pharma companies can barely make themselves yet. So to then go and validate a tool independently to say, oh yeah, I can make that work is not the same as getting a whole load of small molecules. So there's still lots more data to generate, I think.
I'll go for a slightly hotter take. I think we've got loads of data and we're not using it effectively in a lot of ways as well. So I think we have amassed loads of biomedical data, loads of clinical data as well. I think there's lots of ways we're not leveraging that effectively. Of course, also I've seen a lot of the kind of hidden data that we're not using as well today. I think we run small patient focus groups on 10, 20 patients. We extrapolate those findings out to the entire population, and we wonder why people don't behave on our trials as we're expecting them to. That's quite an unreliable thing to do. That data exists at scale. It's just we've not taken the time to actually process that and curate that and put it into an accessible format. So I think I do agree there are different types of data we could collect and different ways we can look at that, but I think we've got a lot of valuable information that we're sitting on and not leveraging effectively.
And Hannah, just staying with you, how Luvida is using AI to improve the clinical predictability?
Yeah, I suppose some of the case studies I talked about earlier in terms of protocol amendments, we're really focusing on as well, reducing the timelines for enrollment. So something that just baffles me is that we ask if a patient wants to be part of a clinical trial. If they say no, we know nothing more about that patient. We don't track that patient. We don't understand why they've said no. There's very little understanding of that. That's a huge amount of people that are saying no that we could make as part of our clinical trial and bring in as participants. That's what's increasing our timelines. Even worse, actually, when a patient drops out of a trial. I know anecdotally, I won't share who, but recently there was a trial happening, and it's a long, several multi-year trial. Lots of the patients, participants wanted to drop out because they wanted to go on holiday. And they couldn't get travel insurance, and that travel insurance was very, very expensive, thousands and thousands of pounds. And so, that's an on-trial cost that then exists. So you can do things to mitigate that up front, right? You can put a list of travel insurers in place for those people. You can know that you're going to need to cover those costs as part of the trials and not be hit with those downstream costs. And you can avoid those patients withdrawing in the first place, because some of them did withdraw because of that. So I think there's a lot we can do to encourage participation in clinical trials, and we're missing a lot of that today.
So one of the recurring themes in drug development is that decisions are often influenced by experience, opinion, or politics, or company dynamics, just as much as data. And it would be, in an ideal world, you would have a more objective way of even just like designing clinical trials. That's who is the loudest in the room. That's don't tell anybody, but that's my experience. That's how sometimes it happens. And it would, so HANA, so again, Luvida relies heavily on AI, and it's very important that when you have an AI-informed, or I think it is very important that you know that you can trust that information, so it's not just another voice in the room, it's not another opinion. And how do you make sure that your client really trusts that information?
Yeah, I think this touches on a lot of things that Leona and Helen talked about as well, and confidence and trust, and we've talked about a lot of the evidence, making that super transparent. I think that's really important. I think it's important to be assessing and evaluating these different answers that are coming out. We do that by using historical trials and seeing whether, based on the data that was available when that historical trial was done, could we have seen it? That's the quickest way to validate something in the clinical trial space, because of course, if you're actually running a clinical trial, that would take a long time to see the outcome of that. So I think really rigorous evaluation on those sorts of things, and making sure that everything is traceable back to the source. Super important for decision making, important for regulators, super important for patients and their safety as well, and then also in getting drugs and treatments to them quicker.
And many times we use the expert in the loop, or there was the lab in the loop. So it's not just a black box, and then I come back and something happens. So it's sort of supervised.
Yeah, totally. I actually don't think that we're at a point yet where we, maybe some people are, but where we should be just kind of going into ChatGPT and saying, build me a protocol, and then using what it gives out. I think we need to still have critical thinking. That's something that, scares me about what we could lose in the future. I think you need to ground all of that in very specific evidence. Those models are going out to the entire internet. There's a lot of misinformation on those. I think it's really important that you're grounding that in the right trial evidence that's being carefully monitored. And I also think it's important that we give all the information, and at the end of the day, a human, an expert, someone that knows this space really well and has experience, is ultimately making the final decision about what's being done. So I think there's things that AI is amazing at, like I've said, these huge multivariate problems. They're things that we're not very good at as humans. We need to leverage that alongside our own capabilities and where we really are clinical experts to make those final decisions.
I think it's understanding where the data came from, what was the context behind that data, and is it going to answer the question you need it to answer, or is it going to be noise, that then generates a delay in your process?
So in a scenario when an AI, system or generates better data than your human team, would you use it for portfolio decisions? So where do you draw the line today? Maybe late in 10 years' time it will be different, but...
I think the analogy that someone used in a conversation recently with me was that it's like driving a car. So the question is who's driving and who's the car and who's driving it. So is the human still driving the car? Clearly the car's getting more advanced all the time, with more power, more data, more analytics. But the fundamental question to ask is, are we still, hands on the wheel, or is it totally autonomous and taking you to the destination like a driverless Uber in San Francisco? And
got them in London now.
Well, apparently, which is a bit scary because London traffic is crazy. But I think that will start to happen. But I still think, in this space with patient safety ultimately on the line, I think it's still going to take quite a long time for us to get to that point. And I think everyone will put a different set of safeguards in place. But I think at least for now, people are going to still want their hands on the steering wheel.
There are companies trying to predict toxicology.
There is. Using the generative AI in toxicology is a reality now. And 2 years ago, actually, FDA had a big 2-day seminar about discussing how we can tackle the generative AI on GLP toxicology. trying to understand, putting the best people from toxicology and AI side, because with the generative AI, it is giving a different answer every time, which is different than the SOPs that we are familiar with, GLP toxicology. So how you will fix something that is different every time, but this is a new way of thinking, and there are multiple good theories. So this is also going to be tackled by creating confidence, while that is going on parallel under the oversight of an experienced toxicologist, which is one example. But in general, we are going through a change in the way we are doing things. So that is AI, that is complex in vitro models. And while we are building trust, there will be human oversight for a long time, or traditional methods will be on the side still during that transitional period. And at the same time, there will be always some people who will not accept any evidence. That is the human element. There will be 10% of the population of your colleagues who will not accept any evidence, data, whatever you show them, because that is part of the change management. But it is illogical to put your resources in changing the minds of that 10%. You need to focus on the remaining 90 and creating data to create the confidence and building the processes so that we can move forward. Maybe 6 years ago, I have been in situations where certain senior leadership will refuse to talk to me if I said MPS and safety in the same sentence, even if it was investigative toxicology or safety pharmacology. Today, things are very different because we have built so much more data and the confidence has increased, and the benefits and how these could be utilized become, the realization of that becomes unavoidable.
So do you see AI generating sort of new insights, or just using the data we already have and help make better decisions?
I think today it's the second one. It's using what we have to help filter out what looks odd, so you can make a comparison, so that the human can make a better or quicker decision. Probably in the future, it will bring its own opinion. How it's learned what that opinion could be is what is the piece that we need to understand more about. How is it validated? How are these systems, environments, what is the life cycle? How are you continuing to make sure it's fit for purpose and generating the kind of outputs that are useful?
Just one more question on clinical trials. What are some of the biggest challenges with clinical trial planning and design?
I'll take.
I'm not looking at.
I mean, it's hard to pick one.
Yeah, how long do we have? Yeah.
Yeah, I think something that I'm really awakened to recently is how much we're kind of lift and shifting things. So a protocol that was used before, we'll take that same protocol and we'll implement that again. And I don't think we've seen, or we haven't seen, a huge shift in some of these. The duration of trials is actually increasing, the cost of trials increasing. But I think there has been a dearth of innovation in clinical trials. That is changing. The last couple of years I've seen a lot of new innovation, and I think that's really cool. I think there's lots of different things that we can try. But I think we need to be open to trying, and I think we need to try and use different techniques. I think there's a huge opportunity for streamlining trials, reducing site footprints, increasing enrollment from patients, reducing patient burden in trials. If we keep taking the last trial that we did and we worked on and using that as our template and basis for the next trial, we're going to keep repeating those mistakes. So yeah, I mean, for me, it's about innovation, thinking differently, and I think that's where AI can actually prompt and help us to maybe make some of those decisions.
I personally, go on.
No, I was going to say it's also designing it to answer the question that you need, not trying to solve the world. I think a lot of problems we have is regulators expect one thing, health technology and payers expect something else, and then the patient needs something else. And how do you bring all of those three needs together in a non-complicated, easy to run study design? And that's the challenge.
Definitely we need more innovation, also AI, post-candidate selection. I think we are at a stage when getting a candidate fast, like the AI-enabled drugs, we can do that. So you can take your drug and answer your question in patients really fast. The jury is still out whether those drugs are better or not. I'm not sure those companies set out to answer that question, that they are supposed to be better, they are supposed to be just faster, but that's just my opinion.
Yeah, there's a bottleneck of drugs now because we can't get them through trials quick enough.
Exactly, yeah. There are a certain number of patients, there's a certain way of running trials. And I'd like to talk about speed versus quality, because eventually sometimes you compromise. And I don't know whether you have any examples when you compromise, was it worth it or you didn't? Quality versus time.
A lot of what the challenge is, you've got to scale as you get through your development pathway. So when you've got your asset identified and you know your target molecules, your manufacturing scale is one size, and as you go further through the clinical development process, you have to scale up and improve that manufacturing process and become ready for commercial batch sizing. And that whole part of the pathway is often done in isolation to the clinical or the toxicology or the other data requirements that are being timetabled, and you can end up having big delaying factors because you haven't got drug ready for the next start point of your trial. So how you can then link all of those by understanding the interdependencies, what data do you need for the next stage of your, and when to start that. So sort of putting that plan, connecting the plan, and making sure you can remove any of the white space will help bring things to clinic quicker and then hopefully to patients more quickly.
That's my day job pretty much. I don't think AI is gonna take that away very, very soon. That's for sure.
I mean, there's a huge amount of areas where time is spent and invested, and it's not compromising quality in any way. It's redundant. I think there's a lot of governance processes and things like that in place that obviously you want the governance checkpoint, but do you really need to wait 4 months for the next governance meeting? And a lot of this kind of dead time that I think is happening. So I think there's huge ways that lots of opportunity for cutting those timelines without compromising on the quality.
Obviously, nobody ever done that compromise quality. So this was a really bad question. I'm just looking at the time to leave some time for the audience. Let me just go to my closing questions. So if we look ahead to the future, there was a similar question before, but I'm going to ask differently. Let's say 5 years, we are looking back today, what would look the most outdated compared to 5 years, 10 years? Because if you look back 5 years, would you think that the FDA would consider no animal tox for antibodies? I think that would be...
EMA would have considered. FDA would have not considered because of the differences in the legal systems. That is also a factor that influences the responses. So potentially, maybe I would say, just looking at the drug discovery and development entirely from technology-centric point of view. I'll use these tools in this order as a pipeline to a decision. We will leave that behind. That will be completely outdated. And we will completely settle on decision-centric way of choosing platforms, which is the phase that we are going, but that will become the norm. So we will start with the question and rank the parameters of what we are trying to solve, what questions we are exactly trying to answer, and what kind of platforms, AI in chemical or in vitro or anything in between, or animals, will be the best suited, either alone or in combination, to answer these questions, and then we will move that way.
I would like to see that we stop thinking about things in boxes of what each thing might offer and look at all of them to complement a sort of holistic, connected evidence generation system, so that we're not just reacting or selecting, cherry picking. It's a planned, connected way of generating evidence that's trust-based, transparent, and suits the needs of the patient.
Are we taking it in turns? Okay. I might be pushing for 5 years, this might be more 10 years, I'm a bit visionary. But personalised trials, stay with me. So at the moment, there's obviously lots of things that you need to be the same on a trial. You need to give them the same treatment. You need to make sure that your randomisation and blinding is good. There's also lots of other factors where maybe some people need a bit more support than others. Some people might need home visits. Other people might need support with getting to the clinic. Other people might need food vouchers. So there's lots of different things that you can put in place, which varies by site and by person. So that's where I see us heading, is being able to support people to take part in a clinical trial much more effectively.
Yeah, I think I'll build on both of those points actually. So I think what Leona said about the connectivity, I think the speed that AI tools are developing is just ridiculous. And so I think the tools that we have now and we're talking about now, I think those will be the thing that are most defunct in 5 to 10 years' time. I think the way that we utilise them, the power that they have, will just exponentially grow and will outstrip a lot of other things. But they, as I said earlier, we'll still need to feed them data. We've still got to feed them quality data, because they're only going to be as good as the data that they take. So, but I think the way that we utilise them and the way that the data connects from the very earliest points of discovery all the way through to clinical development, I think is going to be key. And then I do think there is opportunities for sort of chip-based technologies like our own to produce much more personalised type approaches. Patient on a chip is definitely not that far away, where you can take patient material, culture them in a platform, get individualistic readouts, and then feed that through to clinic. And I think that will, in the next 10 years, start to become. But again, it will be one part of, again, the toolbox. Won't work for everything, won't work for every indication, but it will work in certain circumstances.
Thank you very much. Now I'd like to open questions to the audience. No? Shall I ask more questions then?
Where's the AI questions?
Sorry?
Where's the AI questions?
Yeah, does the AI...
Oh, he's gone.
Oh.
Well, there was some, I mean, I was speaking to Ben before, and we were chatting about the kind of upfront costs of using platforms like NAMs and stuff like that. I don't know if there's anything you wanted to... Sorry, Julia, but you know, anything to do with the kind of, they save time and then essentially money, but what about the upfront costs of adopting these technologies? I don't know if there's any, I mean, it applies, I know we keep going back to AI, but it applies to that as well. Like, you know, bigger pharma companies can afford to kind of sweep up those platforms and utilize them, but when, especially in the one nucleus ecosystem, a lot of our member companies are smaller and just starting out on their journey. So how can they access these technologies? Is that realistic? Just a thought.
It is definitely an issue, and this is something I have come to the realization fairly recently. For big CROs, or like top 10, 20 pharma, adopting these risky technologies or any risky technologies is fairly easier in the sense that they can absorb if innovation is a risk. So if it doesn't work, they have the buffer to be able to absorb. and be the pioneers to learn it upfront. Whereas when we are looking, not even the small biotech, really small startups or companies, they have maybe 3 assets. And they might be the ones working in the rare disease spaces, for example, or something else. They might be the ones who could actually benefit the most from using MPS systems. But for them, they don't have that kind of budget. So their fixed budget is 95%, whereas the variable budget is maybe 5%. So they have to have their must-haves, and then the good-to-haves. And then there is the little bit budget. And when they are looking at whether we should use an MPS system, it is not always, is it because better? They do, even at the times when they do agree it is better, it is more expensive. There's no way denying that. Yes, cost per data point is not that expensive, but the whole could be expensive compared to an animal data. And if for them the choice could be easily, if I buy MPS experiment, if something goes wrong in my toxicology package and I need to reduce something else, I will not have the money. So the question could easily be not because are they better, can I afford to have them? And in that case, it really burdens me that we are getting into a case where there is not equity for everyone to have access to these really useful but slightly luxurious tools. So how we can create equity for the ones who might need it the most, which I don't know yet.
I think though that I think it's 2 things. One of which is like in any market, right? Scale will reduce cost. So if we're honest, right, the amount of animals that are used versus the amount of chips that are used is still like, up there versus down here, right? So as that happens, the price will naturally come down. And I think it's, Penny, you had it on the slide, right? The CROs are the central cog in the wheel, and that there are no CROs offering these technologies just yet. They may be very soon. But they're not yet. And so when that happens, that kind of opens up the doors for everybody. And I think one thing that, and on a very practical point, the models that we build, for example, are with primary human cells. These are inherently expensive. They're a finite resource. We get them from patient donations, which is fantastic. But stem cell technologies should change that, right? And you should be able to generate large banks of lots of reproducible, consistent cell materials. That doesn't happen yet. That still needs considerable innovation so that all the different cell types of each one of us can actually generally produce to have really high quality, really high fidelity. That will happen. The IPS space has come a long way, but there's still, again, it's in its infancy. So all of these things are all interwoven with each other. And then I think the last point about cost is, again, say go over the same point, but if you use AI to design your MPS study, you'll do a better MPS study. So rather than, it's about a mindset of how you design the experiment in the same way you want to design a trial, utilize these tools. And if you use the same tools for your trial and your preclinical studies, then you're probably going to have quite a bit.
We're quite lucky in that we've actually been able to open up our platform to biotechs. We don't charge for it. We work on data sharing agreements, so we're supporting with collecting some of that information around the patients who are enrolling, who's not enrolling, what are their motivations for that. And so actually you can use the tooling, build out a protocol, find those risks, find them early, hopefully save you lots of money downstream, but definitely save you money upfront in that trial design process and supporting through regulatory decision making. So from my perspective, not more expensive.
But it's a really good question. Yeah.
Cool. Thank you.
Thank you.
I think pretty much, that brings us time. Thank you, guys. Thank you.